Hi everyone, my name is Aaron Sato and I'm the CSO of the Twist Biopharma vertical here at Twist Bioscience. And today I'm going to give you an overview of all of our synthetic DNA technologies at Twist Biopharma that enable us to have fast and responsive antibody discovery and optimization technologies. I always say the best companies out there really understand the one thing that they're really good at. And for Twist, that's really to print DNA on silicon. So shown on this silicon chip, about the same size as a 96-well plate in the middle of the slide, is our Twist silicon platform where we can actually print up to 1 million individual oligos up to even 300 base pairs in length. And so basically it's a fantastic technology for us to make highly diverse pools of individual oligos. And we use these oligos to make all kinds of custom DNA products. So custom clonal genes is our top product that we sell. You can go online, order any clonal gene up to 5 kb into any vector you wish, and it's delivered to you within a short period of time. You also can use oligo pools for NGS enrichment kits. So we sell all kinds of custom enrichment kits for sample prep ahead of NGS sequencing, where you can basically pull down specific exomes associated with specific gene sets. And then finally, we also use oligo pools to make high quality custom libraries, which is actually key to the Twist Biopharma vertical within Twist Bioscience. And I'm going to talk to you about that right now. So how do we use oligo pools to build these unprecedented libraries? So basically, the way it works is if you think of an antibody domain as essentially a collection of 3 different loops or CDR loops, we can basically make pools of individual oligos that encode for the diversity seen in each of those loops, but on an individual level. So the reason why in my mind this is really game-changing in terms of how people build libraries is that in the past people have typically used what's called a degenerate oligo to encode for that diversity. So they would either use mixtures of nucleotides or trinucleotides to mimic the amino acid diversification seen in antibodies in nature. But they oftentimes can't get it exactly right. So here, I can actually code for exact sequences of CDRs seen in nature, put them in an oligo pool, and then just shuffle them together in the context of a single human germline framework to create a hypervariable human antibody domain. So this allows me at the end of the day to create an antibody domain library that has that almost exactly indistinguishable from anything that might come out of a human being. I also can remove sequences in these CDR loops up front to remove any kind of liabilities from those sequences. So for example, isomerization, cleavage, deamidation, or glycosylation sites. And then finally, if I choose to, which is actually key to our difficult to drug target libraries we'll talk about later, we also can insert specific motifs into those CDR loops as well. So just to remind everybody, again, Twist Bioscience, through their large-scale silicon DNA synthesis platform, allows us to build these very precise DNA libraries. I'm going to focus a lot on utilizing them to create antibody libraries, which is key and central to the Twist Biopharma offering. I'm going to spend the bulk of the presentation talking about. But I just wanted to make sure that everyone's aware that, again, through our library technology, we can create very precise libraries using the oligo pools that gives us precise base-by-base precision that allows us to control codons. If we wish to, we can actually have multiple lengths for CDRs, in particular for heavy chain CDR3. Of course, we can avoid any restriction sites and then one motifs in our libraries as we build them. It gives us really, again, precise combinatorial control over what CDRs we mix together. We can do ratio-based amino acid distribution, and if we choose to, we also can have multiple germline scaffolds as part of our library. And then finally, at the end of the day, all of our libraries are validated by the next generation sequencing. So now on to Twist Biopharma. So Twist Biopharma, we're a division of Twist Bioscience. We have a small team where we're utilizing all the fantastic DNA products of Twist to enable us to help pharma and biotech companies discover as well as optimize antibodies. So we really sit at the front end of the biologics workflow, where we can help others discover antibodies from synthetic antibody phage display libraries, as well as if you already have leads or have leads generated from our libraries, we can help you optimize them for greater potency, selectivity, and also potentially humanize them if they're derived from a rodent source. So just on a high level, what are all the different areas that Twist Biopharma is focused on? So first of all, we're offering up a whole suite of different antibody phage display libraries that are generally used. They're naive synthetic libraries. I call it my library of libraries, where we have different scaffolds from Fab, single chain, VHH that we can basically pull out of the freezer and use against any target we wish. We were also focusing on utilizing creating libraries around difficult to drug targets. I always say synthetic libraries are actually best used against these types of targets because oftentimes other antibody technologies might have difficulty with targets such as GPCRs or ion channels, and they also may be limited in terms of their epitope diversity. So here, by using a synthetic library and also tailoring that diversity, as I'll show later, we may be able to drug some of these difficult to drug targets such as GPCRs. Thirdly, we created a whole platform for antibody optimization that we call TAO, which stands for Twist Antibody Optimization. It's our technology where we have some very unique software that has a very large human NGS database as part of it. And we couple that with our library technology I talked about before to create a solution whereby we can optimize antibodies by shuffling natural human diversity in that library technology I talked about. And then finally, as part of all of our projects, we make a lot of antibodies. Typically, we make at least 100 antibodies per antibody discovery project that we do. And so we've gotten very good at coupling our DNA synthesis technology at Twist with our ability to make antibodies. And so we're actually kicking off a new alpha product this year to actually enable others to not only synthesize DNA encoding antibodies, but also offer them the ability to also make large numbers, be at small amounts of antibody protein as well. So as I said, our vision for Twist Biopharma on the discovery side is to create what I call our library of libraries. The reason why I like this idea is because, again, in phage display, we're oftentimes limited to the total diversity that's possible in phage display, which is typically around 10 billion different antibodies in a library. To increase the breadth of our diversity of our total antibodies in all of our libraries that we have, one solution is actually just to build more libraries. So for example, if I had a plate of 100 libraries, each one having 10 billion different antibodies, I would have a total diversity of antibodies on the order of 10 to the 12th. So by enabling, because we can build these high quality libraries that I talked about before on the order of months, we can have a huge array of libraries that gives us a much greater breadth of diversity than others have, which again, a lot of other companies have great libraries, but oftentimes they're limited on the numbers because they're just so difficult for them to build. At Twist, since we're so great at making DNA, it's very easy for us to not only make high quality libraries, but also in a very short period of time. So in terms of the libraries that we have today, our workhorse library that we've used a lot in many different projects is what I call our Hyperimmune Library. Our original library is a fully human Fab library that has diversification in all 6 loops of the fully human Fab, which I'll talk about more later. We also have a common light chain version of that same library that we can use for common light chain bispecifics. The second class of libraries that we have is we have actually a whole suite of different VHH single domain libraries that I'll talk about later, which again are proving to be very useful in finding antibodies to almost any target we wish from either these llama-based framework or humanized framework VHH libraries. Thirdly, we have a library that we call the Structural Antibody Library, which is actually a library derived from all known human antibodies that are in the public PDB database, making the assumption that antibodies that have been crystallized are very stable and developable. We sought to create a library utilizing the diversity seen in those antibodies. And then finally, as I mentioned, synthetic libraries are oftentimes best used for difficult to drug targets. And we've actually created a suite of different antibody libraries to GPCRs when we actually have 2 different libraries, GPCR 2.0 and 3.0. We have an ion channel library that can potentially be used to drug these difficult ion channel targets. And then finally, we have a library focused on carbohydrates. In terms of how we typically initiate a campaign using our library of libraries, again, we take our multiple libraries, oftentimes each one having a diversity of greater than 10 billion. We do our selections and screening against that, either recombinant protein or cell-based target. We pick individual clones and do ELISA if it's a recombinant protein target, or if it's a cell-based target, we oftentimes just go straight ahead into picking colonies and doing Sanger sequencing and also NGS sequencing to get all of our unique clones, which we then port immediately into reformatting to full IgG and DNA scale up. Again, since we sit inside of a DNA company, it's very easy for us to make large numbers of DNAs, and then we port those directly into our high throughput IgG alpha product that I'll talk about later that enables us to make hundreds of antibodies that we can then push into binding and functional assays. As I mentioned, we're actually kicking off a new alpha product at Twist this year, which is where we're going to offer up not only the ability for folks to actually synthesize the DNAs that encode for either IgGs or VHHFCs, but also for Twist to actually make them on a small scale, either on a 100 microgram or 1 mil scale, or 8 mil 1 milligram scale. So what we find is that, again, we feel that folks out there are already doing this a lot internally and also reaching their capacity. And so by offering them this ability to not only order DNAs, but also IgGs, we think it'll be really helpful for the biopharma and biotech industry. Okay, so now I'll get on to our case, the libraries that make up the library of libraries. So first of all, our GPCR 2.0 library, I call it our motif-directed library that's focused on GPCRs, and I'll show you now how it was built. So it came from the initial concept for the library came from this paper by Liu et al. Back in 2014, where they took a known crystal structure of CXCR4 bound to a loop peptide called CVX15. And what they hypothesized is, could they take this loop peptide and actually graft it into a bovine IgG shown in the middle of the slide, in the hopes that that grafted peptide would then still remain and continue to bind CXCR4, but now in the context of this long heavy chain CDR3 of this bovine IgG. And that's exactly what they saw. They found that the CXCR4 grafted peptide onto the bovine antibody retained binding to CXCR4 transfected cells, and also retain the antagonist function of the original peptide. So what we sought to do using this initial concept is, could we create a fully human antibody library, not a bovine one, that had a very long extended heavy chain CDR3, much like the bovine case, but rather than just have one motif that bound one GPCR, what if we could create an oligo pool that had over 100,000 different motifs that bound all kinds of GPCRs that are known throughout literature and the patent literature? And so that's exactly what we did. We created a motif-directed library that has a very long extended heavy chain CDR3 and a fully human framework that we can pan and screen against any GPCR we wish. So where do we get all those different motifs? Of course, chemokines that are, again, known ligands of GPCRs are rich in these motifs. So again, we're taking small pieces of these proteins that bind a range of different GPCR targets. A lot of GPCRs also bind peptides. And so again, we took pieces and motifs derived from either helical or loop peptides that are known to bind to GPCRs. Some GPCRs actually interact with themselves through their N-terminal domain. And so we took some motif content derived from the N-terminal domain, which again binds the second extracellular loop, and we put that into the library as well. And then finally, a small portion of our heavy chain CDR3 diversity in this motif loop library also is derived from anti-GPCR antibodies. So there is a fair amount of antibody GPCR content as well. So using all of those motifs, we actually had to do a lot of pre-work before we started using that motif loop library. So we needed to make sure that we had a scaffold that could actually accept these motifs. And so we did a lot of work to look at which heavy and light chain combinations could accept a very long extended heavy chain CDR3. And we used a flag tag as a surrogate for these different motifs. And so after doing about a year's worth of work, we were able to settle on 2 different heavy chain frameworks and 4 different light chain frameworks that show high levels of displayability with these long heavy chain CDR3s with this flag tag. And so once we felt that we had a good framework and loop length to hold and display these motifs, we then put in this large loop library to enable us to create the final GPCR library that we have today. And again, we transformed it on the level of 10 to the 10th, or 10 billion different antibodies. So I'll just show one example of where we used it. So we panned and screened it against a GPCR called GLP1R, which is a class B GPCR. It binds a linear helical peptide called GLP1 peptide. And so we found a whole panel of antibodies by panning our GPCR antibody library against GLP1R overexpressing cells. We found a whole panel of different antibodies that bound to GLP1R expressing cells with high affinity. And one of those antibodies called TB01-3 at high concentrations, shown on the left, actually showed significant inhibition of cyclic AMP signaling. So shown as a titration of GLP1 peptide in black, where you see a nice dose-dependent titration and activation of cyclic AMP. in this DiscoverX assay. And then the green line is actually if we add a high concentration of the antibody and do the same GLP-1 peptide titration. And again, we see a shifting to the right, indicating that we have inhibition of signaling. In the middle, we see that if we do a dose titration of the antibody now at a constant concentration of the ligand, we see now a dose-dependent inhibition of GLP-1R signaling at high concentration. And then finally, we did a PK study and an in vivo efficacy study with the antibody showing that it actually not only shows a long half-life, but also shows an expected efficacy in a glucose tolerance mouse study. It's shown here up in the top right. The other GPCR library that we've created, unlike the motif-directed library I just talked about, is our library that we call GPCR 3.0. So here, rather than create a motif-directed approach, we basically took all known GPCR antibodies that we could find. So we found over 61 patented GPCR antibodies that bind over 22 different GPCR targets. And we took those as an input data set to create the library I'll talk about now. So it actually has 2 different heavy chains and 2 different light chain frameworks. And we basically took all the diversity from those different antibodies and encoded a whole series of oligo pools and created a library based on those frameworks I just talked about. Shown here is just one example where we discovered an antibody TB83-107, and it found that it's actually a very potent CXCR5 antagonist. You can see on the left, we see a nice dose-dependent binding FACS titration curve for binding to CXCR4 stable transfectants. And then on the right, you can see that the same antibody in orange actually shows, again, a shifting to the right or an inhibition of cyclic EMP signaling in response to the known ligand of CXCR4, which in this case is CVXL13. Now I'll move on to our Hyperimmune Library series. So again, our Hyperimmune Library is again a fully human Fab library. It's based off of a single human germline scaffold, VH3-23 VK139. Here, I wanted to create an unprecedented library that no one else had ever built before. And to do that, I started with, again, the idea that, the primary determinant in antibody binding is heavy chain CDR3. And so I wanted to create a very diverse, a library that had a very diverse heavy chain CDR3 oligo pool as part of the main part of its interaction with its antigen. And so we combed through our human NGS databases, collected together over 2.5 million heavy chain CDR3s derived from both human naive and memory B cell diversity. And we encoded that into the heavy chain CDR3 position of this fully human Fab library. We also put in diversification in the other 5 loops, but the main part of the diversity in this library is all mostly directed in the heavy chain CDR3 region of the antibody. And then again, we put that into our Fab phage vector, and then again transformed it on the level of 10 to the 10th. Shown here is just a little bit of data from using the library, where we are actually able to find several human cyno cross-reactive clones to CD3. Those of you that are familiar with this target know that it's actually very difficult to find human cyno cross-reactive clones. You can see on the left and on the right, we have 2 clones that have double-digit nanomolar binding to heavy chain CDR3, to CD3 epsilon. We've also used this we've also used this library for finding antibodies to SARS-CoV-2. And so shown here is just a little bit of data where we've actually found several antibodies, shown here TB182-3, 4, and 7, that have high affinity binding either to the S1 monomer or the S protein trimer on the order of picomolar to nanomolar affinity. So again, showing the power of this library to find very high affinity antibodies to all kinds of different targets, and in particular to the SARS-CoV-2 S1 protein. And we've gone to show that several of these antibodies show very potent inhibition of S1 ACE2 interaction. Again, many people know that, again, this is the main target of therapeutics against SARS-CoV-2. And you can see on the top right, we're trying to find antibodies that not only block the S1 protein, but also potentially ACE2 to block infection of the virus. Here is a cell-based assay that we've rigged up where we have Vero E6 cells which express high levels of ACE2. We come in with S1 mouse FC as S1 RBD mouse FC as a surrogate for the SARS-CoV-2 virus. And then we look to see whether we can block that interaction with the cells using our antibody. And you can see we have a number of different antibodies, again, derived from the Hyperimmune Library, where we see very potent inhibition of SARS-CoV, of the S1 RBD binding. So again, I've just gone through the construction of the Hyperimmune Library. Now here's a couple examples of how we've actually discovered antibodies from the Hyperimmune Library. So shown here are just one example where we've discovered antibodies to CD3 epsilon, which cross-react between human and cyno CD3. And as you may know, finding antibodies that cross-react between human and cyno is actually really difficult against this target. And so shown here is just two examples of two antibodies that we found that have decent levels of cross-reactivity between human and cyno CD3. We also use the library to find antibodies to the SARS-CoV-2 S1 protein. So shown here are three antibodies that have either nanomolar or even picomolar binding to S1 and or the S protein trimer. Again, showing that we can pull out very high affinity antibodies against any target we wish. We also then created a cell-based blocking assay for S1. Again, we started with VRE6 cells, which express high levels of ACE2, and we use S1 RBD mouse FC as a surrogate for the virus. And what we show is we can find a whole panel of antibodies that block the interaction between S1 RBD and the ACE2 on the surface of these cells. And in particular, TB1A2, 3, and 4 that I talked about before have high levels of competition with S1 interacting with ACE2 on the cells. Now moving on to our VHH libraries. We actually have 4 different VHH libraries that I'll talk about now. They basically started with over 3,000 different VHH sequences that are derived from a database that we found that all had known antigen specificity, and we use those as an input data set to create the 4 libraries I'll talk about now. So the first library is what we call our VHH ratio library. It's based off of a consensus llama framework. And we basically created synthetic oligos for each different CDR loops to mimic the diversity seen in those 3,000 VHHs. So very similar to a degenerate oligo like I talked about before, but in a much more controlled way where we're actually allowing for specific ratios of amino acids shown here on this slide. The second library is what we call our VHH shuffle library. So again, on a consensus llama framework, but rather than make ratios of oligos that encode for what potentially could be seen in this diversity set, we just use the exact CDRs directly from the 3,000 VHHs in the database and just shuffle them in different contexts to give us antibodies that have unique specificities to human targets. The third library, we do the same exact thing, but we do it in the context of a humanized VHH framework, so we call it our VHH human shuffle library. And then finally, we took that VHH human shuffle library one step further and replaced the CDR3 diversity derived from llamas in the VHH human shuffle library, and we put in the Hyperimmune heavy chain CDR3 diversity oligo pool that I talked about before to give us a final library that has a humanized framework and human heavy chain CDR3s in the CDR3 register with only llama CDRs and CDRs 1 and 2. We've used this library to find antibodies to TIGIT, which is an immuno-oncology target, where we find either double-digit, triple-digit nanomolar monovalent affinity binders from all these different libraries. And we've also used these libraries as well to find antibodies to the S1 protein from SARS-CoV-2, where we find either nanomolar or maybe picomolar binders to both S1 as well as the S protein trimer. And again, similar to the Hyperimmune case, we found a whole panel of VHHs that also inhibited the binding of S1 to ACE2 on VRE6 cells. I'll finally move on to our last library, which is our library that we call our SAB library, which again is as I talked about before, is based off of this database, which has all of the known human antibodies that have been crystallized. And again, we use that as our input data set to create this fully human antibody library based off of those structures. And again, it's built off of a VH3-23 VCAP 139 fully human germline scaffold, very similar to, again, the Hyperimmune scaffold that I talked about before. But again, now we're shuffling CDR sequences derived from those fully human antibodies that are in the PDB to potentially give us antibodies within this framework that can be very developable because they're derived from that initial input data set. And again, similar to the POC studies I've shown before, we can get antibodies that cross-react between human and cyno CD3, again showing the power of the library to pull out these very rare antibodies that can show this very fine specificity. In terms of coming attractions of new libraries coming up, we're creating a library in collaboration with another company called Natural Antibody, which is going to be an SCFE library based off of antibodies in the public patent database. And we've already done some initial POC studies with it and shown that we can find antibodies to TIGIT, CD3, and even SARS-CoV-2 S1. So again, really a powerful library that we think could have a lot of applications. The next library is a library we're constructing in collaboration with DeepCDR, which is an AI augmented library designed from knowledge of DNA rearrangement, somatic hypermutation. So it's a really fantastic, incredible library where we're designing it from the ground up from the DNA level. So again, it really shows the power of our technology to utilize DNA to build these fantastic libraries in collaboration with a fantastic company that knows a lot about antibody sequences in DeepCDR. And then finally, we're working with a company called Taurus, which has a lot of knowledge of cow antibodies. And so we're trying to create a human SCFE library that's now in an antibody framework that incorporates a lot of these long, heavy chain CDR3s derived from cows that may allow us to drug a lot of difficult targets like GPCRs and other difficult to drug targets. Finally, I'll just end with our Tau platform. Again, Tau stands for Twist Antibody Optimization, and it couples our library build technology with this very unique software that we have. And basically what we do is we use this software to input an antibody derived from any source. What the software does is it looks at your antibody within the specific framework that you set, and looks at heavy chain and light chain CDR sequences in the database and compares that back to your input reference antibody and looks for sequences that are similar within a defined mutational space that you set. So for example, oftentimes I'll use a mutational distance of 3. So it'll basically give me back all CDR sequences within that germline family that are within 3 amino acids of my parent reference antibody CDR sequence. And then I can use those sequences to then construct a library in phage display and do all of the downstream selection and screen to find optimized variants of my reference antibody. And so again, just on a high level in terms of how it works, we put your antibody into the software. It gives us a whole series of oligo pools to make. We order those oligo pools, print the library. We then put that into phage display and then do all the downstream panning and screening to give you your optimized candidates. And shown here is just one example where we did this and increased the affinity of a PD-1 antibody by over 72-fold. So just to conclude, ways you can work with Twist Biopharma. First of all, we're very open to licensing all the libraries I talked about today, as well as doing partnerships around finding leads derived from these libraries. We have a fantastic team that can utilize these libraries very quickly to find antibodies to any target you wish. We also can do partnerships around our antibody optimization platform, where we can again take an input antibody from you and create a library focused around it and do all the downstream panning and screening to find optimized candidates. We're doing a lot of POC work, as you saw from our library, our new libraries that we're creating. And so oftentimes we're finding a lot of leads derived from those libraries. And again, we're very open to partnering and licensing out those leads because we're not a full-fledged R&D company quite yet. As I mentioned, we have a whole separate library team where you can go and order your own custom libraries. A lot of times, many of our partners that order these custom libraries don't have the capacity to screen them. So oftentimes they'll partner with us to pan and screen them in phage display. And so if that's of interest to you, then you should definitely talk to your library sales SynBio professional as well to see if that's a possibility. And then finally, as I mentioned, we're trying to kick off a new alpha product this year for high-throughput IgG. So if you have a need for making large numbers of IgGs as part of your workflow, definitely talk to your SynBio sales rep. And that's the end of my presentation. I'm going to be happy to answer any questions and walk through any of the materials I talked about. Again, super excited to be here today and would love to address any questions or feedback you might have about the presentation. And thank you.